Rolling.
All right, this is a eukaryotic cell. This is going to be a random review video about endomembrane system and cell signaling and gene expression and all stuff rolled into one. So here is the DNA in the nucleus. And let's say we have a target gene in question, which I am going to highlight with this orange box. Here is where that gene is. And yes, Joyce, I appreciate you kind of zooming in the best you can. Let's say that there is a hormone signal. Hey, how are you, Dr. Webe?
I'm good. Are you gonna not come to the department meeting because you're running a study session?
Yeah, sorry. Thank you for the reminder, though.
Not a problem.
I'll be there. I might be able to be there soon. This is going to whoop, and whoop, went right into the cell. And here is the receptor for this ligand, this signaling molecule. And now it's going to bind to the receptor. So the first thing, hi.
Hello. We just have a quick question. Can you say?
Yes, I can, if you put it down. What can we tell about that signaling molecule? What do we know about its properties? Joyce, you can answer if you want.
I don't know.
I can tell a few things about the makeup of this signaling molecule because of how it's behaving.
Say that one more time.
Because of how it's behaving, I can tell a few things about the makeup of this signaling molecule. Nick?
You can tell that the molecule is a small non-polar lipid because it goes through the membrane without any aid and binds to a receptor inside the cytoplasm.
A small nonpolar lipid that is a signaling molecule like this is probably a steroid hormone. This is the step of reception, right? This is the receptor. And then let's pretend that this step of reception causes a sort of a signaling cascade. So remember, when a receptor, this receptor happens to be inside the cell, but when a receptor gets activated, it probably has a shape change. So there's the activated shape. And it's going to cause, I'm making this up, the details, but it's going to cause a molecule to bind to it, which will then become active and activate another one, and then activate another one, and then activate another one. This step right here, of course, is what? Transduction. Good. This is the transduction pathway. Now, I don't know if you're going to be able to see this in the video, but probably each one of these is phosphorylating the next one. So that is a little phosphate group being attached to each one. Remember that it is a kinase protein that phosphorylates other proteins. If you are a kinase, you are adding phosphate groups to other proteins. And let's say that this right here is a transcription factor. Transcription factor. I'm talking about that right there. What do transcription factors do? They bind to DNA and allow what molecule to attach and do transcription?
DNA ligase.
No, not DNA ligase. DNA ligase is going to repair breaks in the DNA. So, the DNA is broken, DNA ligase seals it back together.
Sorry, okay. I thought enzyme. Sorry. You said molecule?
Joyce, let me just think about whether I'm going to accept this apology of yours. Apology accepted.
Okay.
What is the molecule, the enzyme that binds to a gene to initiate transcription, to do transcription, as a matter of fact?
Something polymerase.
Yes, something polymerase is correct.
DNA?
RNA polymerase.
Oh, God.
So now that the transcription factor is there, RNA polymerase can now attach. There's RNA polymerase. And it's going to make, I'm going to make this nucleus a little bigger, just so I can have a space to draw this. It's going to produce an mRNA strand from this. There's the mRNA strand. Joyce, can you tell me why, after this is processed, the mRNA strand is shorter?
Because the introns go away.
The introns are removed. RNA is going to go through a pore in the nucleus, and it's going to go out to a ribosome, right? And I'm going to do the ribosome as a red dot. Now it's attached to a ribosome. There it is. And it's going to do translation. So notice that it was a signaling pathway that activated the gene. So this is the response to the pathway. All right, it's a little busy in there, so I'm not going to write that in, but that would be the response. So reception, transduction, response in the nucleus is turning on a gene. And so now we've got our RNA processed, the mature mRNA is out in the nucleus. Let's say that this is going to make a protein that needs to be excreted from the cell. It needs to go outside the cell or maybe into the membrane, could be another place. What kind of ribosome has to make a protein that has a specific destination? What is the ribosome going to be attached to? This is a cell part, an organelle I'm looking for. No, but close. Not the Golgi.
Reticulum.
Yes. This would be a ribosome that's going to need to jump on to the ER, and that's where the rest of translation will proceed, because it's going to spit out the protein, and here's the protein. Right now, I could pause here and talk about the 4 levels of protein structure, right? If this is the amino acid sequence, right, this is first level, right, these are the covalent bonds in between each amino acid, right? Second level of protein structure is this is going to fold up either into an alpha helix or a beta sheet, and that is because of hydrogen bonds that are like here. These are polar attractions. And then third level, this is now going to fold up into a more complex structure, and that's a many bond types. And then fourth level, this polypeptide is going to link up with another polypeptide, again, due to there being many different bonds. Anyway, that's a brief review of the 4 levels of protein structure, but now we have a finished protein and we need to ship it outside the cell. So where does it need to go next after the ER? It needs to go into a vesicle, that's correct. So we're gonna pinch off a little bit of this membrane, and now our protein is in a vesicle. There it is. This is the RER. This is the vesicle. And where's it going next?
To the membrane.
Not yet. It needs to make one more stop. It needs to be like packaged properly.
Oh.
The Golgi. Here's the Golgi apparatus up here. It's like another folded membrane. And we are going to do, the vesicle is going to kind of release the protein right there into the Golgi. There it is. It's going to get properly packaged, shipped off in another vesicle here. And if this is going to be released to outside the cell, if that's the destination, that vesicle is going to fuse with the membrane like this. And then that will open. This is a process called exocytosis, right? Exocytosis. Other destinations this protein might go. It could go into a lysosome if it was a digestive enzyme. It could just be into the membrane. It could become a channel, right? It could be inserted into the membrane there and be a channel or a pump or some type of receptor. If it was a freely floating enzyme in the cytoplasm, it probably would not be made by an attached ribosome. It would be made by a free ribosome, and it would just be in its destination, the cytoplasm. Okay. We've talked about a lot here. The little review of cell signaling, phosphorylation and the importance of activating proteins in a transduction cascade, a little bit about gene expression and RNA polymerase's role, transcription factors' role, RNA processing in eukaryotes, and then about the endomembrane system and protein shipment and transport. So, and protein structure. So, yes, let's stop this video before it gets too long.